Sweet. Well, thank you all for joining me. This is a very special day. We're kind of going over the last year of work. We've probably gotten 10 years of work done in a very short amount of time, but we're very excited to share with you. I have a lot to go over, so I'm going to try to limit how much I talk. Scientists like to bore people, so I don't want to be that guy. I want to introduce our next speaker. We are bringing in Dr. James Brown from Arizona State University. James works in a really interesting space. Not only does he work on trying to make items that would replace plastics, but he also makes sustainable green replacements for things like PFAS chemicals. So things that we have a use for, but end up kind of having this bad impact on the planet. He's an incredible chemist, really excited to bring him all the way out here from Arizona. July 27th, we have Louise Roy. We've all heard of PFAS and the contamination in Maine. We know how big of a deal that is. Louise is actually a former intern of the Institute, and she'll be speaking to that. And then on August 8th, we have Rosie Seton, who is the stranding coordinator at Allied Whale, has been a longtime supporter of the Institute, and has some really cool and interesting stuff to share about the impact that they have. To give a little background about myself, I moved here from Arizona with my wife, who's in the back, Danny. We only got here about a year ago, and so I went from holding snakes to holding lobsters, totally natural. We moved here with our two older cats, who are both doing quite well. And then we've made, a kind of habit of adopting other people's dogs. So these dogs actually belong to Jill, and they're now ours. And she didn't have a choice in that. But we really enjoyed our time here. I've had a really interesting academic path. I've done a lot of really fun things over the years. I used to work with killer whales, where we collected their poop using a dog, and I can tell you about that sometime over a beer. Was lucky to do a TED Talk. I was very involved with politics in Arizona. They would tell me to go to speak to the politicians and to take it seriously, so I wore a shark outfit. All sorts of weird photo shoots. I was in this promo that won Emmys, and I always like to show the next picture because people think science is kind of dorky, but I got to meet Jason Momoa. He is really muscular. His back, I put my hand on his back, and it was just, I couldn't tell if it was like his abs or his back. It was this really impressive guy who does a lot for the environment. So that was a lot of fun. Always like to pay tribute to Susan, a really wonderful, incredible person. I probably don't have to tell you much about her. I would like to share this story. During Susan's memorial, the morning of, I think Cynthia had a little bear show up and scratch her car. Little Bear was the name of a boat that you had together. Yesterday, we were driving back from Bangor and we saw a bear. So I think Susan's here. She's here with us daily. One thing I like to take from Susan is she wasn't a follower. She was a leader. She was a trailblazer. So we're not following her footsteps, as I've said before. We're taking it a step further, and we want to honor her by being that trailblazing institute. And I'm really happy to share with you today what we've kind of gotten done up to this point. I'm glad there's a clock on the screen, so I don't have to talk for 3 hours. Hi, Susan. So you're going to hear kind of a mix of efforts. We do a lot of local work, and then we do a lot of bigger picture work. So really big in Maine is sort of the monitoring of contaminants, excess nutrients, and harmful bacteria. The contaminants come from a bunch of different sources. We talked about PFAS chemicals before. If you have excess nutrients that come from land and end up in the ocean, we can have these phytoplankton in the area that can release a toxin. It's not good for us. It's not good for the seafood. And then there's also fecal pollution that comes from land. These are all things that we're monitoring on a pretty active basis all around the area. Probably locations that you're pretty familiar with. We like to have it be areas where there's people swimming or that's near aquaculture farms. That's sort of priority to us. And we sample regularly. We'll put out these reports that kind of depict the state of that area. So if someone wants to go with their family, they kind of know to stay away from certain places. I just forgot to mention, we are getting a new website, which was very exciting for us all. And this will be a lot more interesting looking when that happens. But this requires quite a lot of work. There are 2 full-time staff at Shaw Institute, myself, Michelle sitting at the computer. We have 3 part-time staff members that are all incredible, but we need a lot of help. So we have wonderful interns with us today. We have a really great partnership with Colby College, who we collaborate with on many levels. And every summer we receive a really amazing batch of interns. We always think it can't get better, and it does. Our interns are sitting right here. This is Ella, Eugene, and Autumn. And this was kind of their crash course into the research that we're doing. A lot of it's a really dynamic position where it's a mix between doing lab work and field work, which is really great because science is chaos. it's never going to be something that's consistent. It's always just all over the place. So it doesn't always vibe with everyone, but they're doing a really great job. Here's a nice action shot here of Autumn doing some wind analysis. And here's Ella, really these majestic pictures we've gotten so far. I'm very happy with this. I covered this one up because people were eating. We got to participate in a necropsy of a seal. This isn't something that we advertise for the position because you never know when something might happen. But Allied Whale ended up having a seal it passed away. And I'll get into it later, but this is a wealth of information with each. It's really sad when an animal passes, but they're really amazing indicators of environmental health. And so it's an opportunity for us to get our hands dirty. And I was very proud of them. It doesn't smell very good. And actually someone that worked there passed out. So they all did great. That was a lot of fun. And then here's another action shot. Do you remember when we used to get sun? Isn't that weird? God, I don't even remember what that's like. Anyways, they've been amazing. They're all incredible, and it's just so much fun to work with them. And it's not just work. We went on a whale watch or a puffin watch and a lighthouse tour. That was really fun. We got some great images of puffins, which are the cutest little sea pigeons, and saw some, again, really great comparison here between a harbor seal and a gray seal. That was a blast. And then this really iconic picture of a bald eagle. I joked about it. I wanted to get the picture when the wave, and it happened. So very, very happy about that. And we've also, again, they've kind of taken our nature of adopting dogs that aren't theirs. This is Barley and Autumn. He's going to become our new mascot, which I'm totally fine with. And then we got to take him to all the most wonderful places to eat that you all know quite well, like Bagaduce here. Back to the marine mammal work. The Institute's been doing marine mammal research for many, many years because they are great indicators of environmental health. This involves us going and collecting animals like this harbor porpoise, participating in some sort of analysis of what the state of the organism is. But this is all done in tandem with Allied Whale, who we were kind of given as a volunteer opportunity for us. So for many, many years, we've done contaminant analysis with marine mammals, and that's going to continue. Last year, as soon as we got here, it pretty much hit right away. We ended up finding a baby harbor seal and looked very sad. I worked with Rosie from Allied Whale, who's going to be giving a talk. And we brought it back. He ended up being much happier when he was in the rehab facility. His name is Pepe. I'm always nervous when they name animals that are in rehab, because if it passes, it's gonna be the worst day ever. But Pepe ended up doing really well. And so we got here in May, I should say, of last year. And by June, all of this had begun. So it was kind of chaotic for us, but we ended up doing sampling for 4 months relating to all of those things that I mentioned with multiple organizations, lots of tests, lots of phytoplankton counted. And if you remember Pepe, he ended up being transported out and was released. And now this was done on Facebook Live. And I had to speed this up. Pepe took forever to get in the water. And at first I was really supportive. And then I was like, all right, Pepe, dude, get to it. This is, I got stuff to do. One thing that's a very prominent part of my background is this scene. And this was sent to me by a collaborator of mine. I forget where, but this is always really interesting for me to look at, because there's a lot of things here that should be there and a lot of things that shouldn't. A lot of colors that shouldn't be there. It looks pretty, but it's not. This to me says we've got a lot of fragmented plastics. These circular ones are actually called nurdles. Those are pre-production pellets that they melt into a mold that's kind of like this. So these are just being spilled at some facility in the area and ending up going in the water or going in the sewer and then back into ecosystems beyond. So this is a big part of how I got into marine research. But the question is, where does this stuff come from, and then what are we going to do about it? Well, we have the process of something big being exposed to degradative factors such as UV light or physicality of the ocean or salt. This water is very salty. We have the shedding of clothing that ends up turning clothing into microfibers that go in the air in our lungs. We have those nurdles that I mentioned that just end up right in the ocean. And then we have what were banned by President Obama, but you have these exfoliants that are in face cleaners that end up going into the ocean. These are all pretty big deals, but we're still kind of on the cusp of understanding how and why the impact is what it is. If it's created to be small, it's called a primary microplastic. That face wash, those are all created to be small. That's the purpose. But a lot of times it goes down the sink. It goes into a wastewater treatment plant and then ends up in the environment. You also have secondary microplastics, which is the result of something fully intact that ends up, again, being exposed to some sort of degradative factor, fragments over time, and ends up in the environment. Both of these can kind of act like, you heard about mercury, DDT, things that can concentrate as it goes up the food chain. But again, when I started my PhD, it was still kind of unknown as to what this impact actually was. More and more research started coming out. Plastics are either hydrophilic or hydrophobic. They like water or they don't like water. There's also a lot of really bad things in the environment that like water or don't like water, and they are attracted to each other. Things like bacteria, phytoplankton, persistent organic pollutants, pathogens, they all essentially adsorb to the surface of the particles, some up to 1 million times higher than what's found in the surrounding environment. So you have the physical threat of it itself. It can be a choking hazard to small animals. You have the internal chemistry of the polymers, something like PVC piping, VCs, vinyl chlorides, a carcinogen. And then you have the adsorbed surface threats from all of these things. And all of that's just like a trifecta. And now we're getting into the nano world. So the smaller they get, the kind of bigger deal they become in the environment. I can't escape poop, right? Killer whale poop, and I worked with wastewater treatment plant poop. So it's just poop. I was a poop guy, which is fine. Did a lot of work in wastewater treatment plants, and we actually looked at kind of sewage sludge as a source of microplastics, because again, that's your house. It's what's leaving your house and then going into the environment beyond. Did a study on looking at all the different types of plastics used or found with wastewater treatment plant sludge. That's the solids that leave the wastewater treatment plant. And then I looked at how they analyzed them. A couple things kind of stood out to me. First, fiber, fragment, sphere are all, you can kind of understand what those are. Flake, ellipse, line, like what the hell is the line? Glitter, okay. Big, diversity of terminology, which kind of throws me for a loop. The other thing that you'll notice out of all these types of analysis to confirm whether something small is plastic or not, visual, because I can just look at something and tell what it is, because I'm Superman. This was a problem too. And this is something that's seen quite often in the plastic world. So one of the first things I did when I got to Shaw Institute was purchase this instrument that's capable of analyzing a tiny, tiny particle and extracting a wealth of information from it. And it's been really, really beneficial for us so far. In the future, if I do a lab tour, we'll show you and have the interns show you how it works. It's a lot of fun. The more I heard about, I got to remember to drink water, I'll pass out. The more I kind of look at the logistics of what the EPA regulates in terms of contaminants and public health, the more I kind of got concerned about this. It's like being a detective. You put things together, and then as soon as the puzzle pieces start to form something, it's very concerning. We know that there's these nasty things in the environment, from plastic to PFAS, fecal bacteria. We do some testing, and then we find it in things like bivalves or in the ocean, the water column. And then we're finding them in top-tiered predators. And so far, this has been a kind of linear relationship, and everyone is happy with this. But then my question is always, what about us? Are we impacted by the same contaminants? Are we impacted by the same threats? When it comes to plastic, that became a resounding yes, but how and why? One of the most exciting projects that's come about recently came about because of these superheroes. Dr. Whitney Cowell is a pediatric researcher that looks at the effects of environmental contaminants on children and babies. Dr. Kanan is one of the most cited researchers in the world in his space, environmental toxicology. Dr. Kanan and Susan Shaw go way back. They worked on some incredible projects together, Michelle as well. And Dr. Trisand is an MD who is a pediatric doctor. These are all massive names at NYU, and they're giants in their space. And one thing that was kind of on the back of their minds that then came to the front of their minds, it's what's the relationship between microplastics and placentas? So we end up having these conversations. Dr. Kanan actually came out to give a talk. And after the talk, he emails me and he goes, hey, Charlie, do you know anyone that has done anything with human tissue? And yeah, in my mind, I was like, yeah, me. Keep going. Do you know anyone that can extract and analyze it? Yeah, his name is me, and I can do that. So we end up having this conversation. And one of the reasons why they're so concerned about it is because a lot of initial models have shown with rats that there can be movement of microplastics from the maternal lungs to the placenta and even the fetal organs. This is the kind of beginning of a research project. There's all sorts of implications with this. They've seen the accumulation and the transport of it up the placenta. So it's traveling into the placenta, into the fetus, enough for research to then extend into the human space. So we start to get these human studies of late. One thing I'd like to point out is the sample size. N refers to the number of placenta samples that they digested to see if there's potentially plastics in them. This first paper went through 6. This one had 2. This one had 17. This was obviously a kind of an alert for me because we do these things called power calculations. How reflective are two placentas of the population? If I ask Ian here about a restaurant and he says it sucks, is that reflective of Blue Hill? Probably not. So I need to talk to more people, right? This is one of the first things that kind of caught my eye as being a little bit weird, but okay, let's keep reading these papers. They all were focused on digesting placentas in search of microplastics. This is our lab downstairs. You'll notice we have these two awesome hoods here. What they do is they allow, if I'm doing something with nasty chemicals and there's some weird volatilization of something in the air I don't want to breathe, I do it in here. I can turn on the fan, it pulls everything up, I won't die. It's a great day. The other thing is that you can imagine there's a lot of plastics all over the place. If you look at the sunlight coming through your window, you see things floating in the air. Some of that might be plastic. If you put this down, this is a plastic shield, funny enough. If you put that down, it prevents a lot of airborne contamination, which is really, really an important thing to control contamination when you work on plastics. I want to bring up a piece from this paper here, which is kind of the godfather of all of these studies. Since the experiments were conducted without the use of a flow hood, the plastic fibers found in the samples were not considered in the results. To me, this says they basically did the research like here, which is never happened. I've never read a paper where that was okay. In the plastic world, you always have to do things a certain way to reduce contamination. How do you know it's not coming from you? Think about where the placenta samples came from, right? An NYU hospital. What is that chock full of? Plastic. So how do you know it's not coming from that? So these papers were very concerning to me. And what generally happens in science is that they get so amped up over something, they just dive in and start publishing. We wanted to kind of set the bar a little bit higher. We wanted to be the leading example for how this work should be conducted. So we agree on this partnership with NYU. And in theory, they're going to start sending placenta samples to Blue Hill. NYU is sending samples to Blue Hill for us to analyze here. I still pinch myself that this is a thing. And so our first aim became to look at characterizing microplastics from placenta samples. But what I did is I said, NYU use a lot of plastic when you do these bursts. And then, yeah, we use a lot of plastic. And I said, can you send me some? And yeah, sure. What do you want? All of it? Everything? So they literally sent me every single plastic item used during a birth, every single one. And each constituent has all these other components. So I was hammering them and getting all these different pieces. It ended up being about 175 things I had to analyze and put in a database. So we now have a contaminant database. So if I find something, I can say it's definitely not coming from the hospital, which is a big advancement of this field and something I'm really excited about. I realized something very, very quickly. This is going to take a lot of money and we need to start looking for it. So we started focusing on kind of a local grant. There was a grant offered at NYU, just in this same vein for a research project that's starting and getting up and running. It's not a lot of money. So we ended up submitting this grant and the max award for the grant ends up being $40,000. But the intent was once we develop this further, we will apply for 2 NIH proposals. NIH proposals are among some of the hardest to get in the entire world. It's extremely competitive. I will say this first grant, we got fully funded, and that started the process of us looking into this further. And I wanted to give you a little bit of background about this project. This is going to be what carries us into the future. All the projects here are important. This is going to be the one that puts us on the stage with the people that are really going to recognize how important Blue Hill is and how important the Shaw Institute is. This is what it looks like. It's called the CHES. So it's NYU Children's Health and Environmental Study Group. About 3,000 women are involved. There's complete life histories involved of everyone that participates in this program. They have certain logistics that they kind of work out. There's a really diverse group of people, but it's amazing, right? How many organs in your body do you just throw out? It's really, really unique to get that snapshot into someone's physiology. So it's an incredibly dynamic program. And we just started diving and I got some beef tissue and some pork, something that mimics a placenta. Basically what I do is I create a chemical mixture of something that's going to digest it using heat to accelerate the process. You end up turning this kind of beef sample into a liquid, and the liquid we end up filtering through these small filters, and then the filters are sent off for analysis. And I'll talk about it in a second, but this is what it looks like. And this is done downstairs. This was not the test anymore. These are actual human placenta samples that we're digesting. What you do is you end up filtering it through this one unit, and you end up with these small filters. So the filter size is pretty big. This is the actual area that it passes through. Again, with our collaboration with Colby College, there's a professor there with an incredible instrument called a Raman spectrometer. The one I have downstairs is 20 grand. This is $450,000. So a little bit nicer. And we created this incredible new method to be able to automatically scan those filters, and anything that comes back as a polymer, it'll tell us. And we can get a really good distribution for what's on the filter. An amazing partnership. This took many, many, many weeks for us to go back and forth to figure out, but we're at a really good place now. But we felt like this is significant. We're setting the bar higher, and that's really important for the field. We ended up submitting this to a very, very competitive conference, which I probably should have picked one that was less competitive, but that's okay. So it's an international conference for CTAC. And this was really special for me because I have a history with CTAC, as did Susan. And it was in Dublin, Ireland, and it was online. Now, it was so competitive that they were turning people away. But not only did they accept our poster, but they gave me a platform to introduce the talk, to advertise for the actual event. And this poster is at the back if anyone wants to look at it. I actually thought I was going to get rejected because we didn't have that data. Like, oh, we found this many plastics in a placenta. It was just new partnership. We're stepping the game up. We want this to be the gold standard moving forward. But again, it got accepted. And it was really fun for us to be able to go and kind of experience this Michelle and I, at the Cliffs of Moher. But already it's pretty amazing that we're going internationally with this, but that's just the beginning. This was aim 1. Aim 2 was to start looking at that life history information. What contributes to someone having a higher concentration of plastics in their body? Now, this amazing cohort at NYU collects all of this incredible data, the kind of normal stuff that we're used to between age and diet, but also, do you use salt, spices? Do you have drug delivery systems in your body? Do you use a retainer or whatever at night? How often do you wear masks? This is very conclusive. Now we can actually say, this type of lifestyle factor might contribute more to someone having higher concentrations of plastics. Let me take a step. Back to the NIH proposal. We applied for an R21 and we're going to apply for an R01. The reason why we're applying for these really amazing grants is so we can, again, step it up even further. And our third aim is to actually look at urine samples from the same cohort to now look at plastic chemicals that were found in the same women. Now, we took it up a notch already. This is like 3 or 4 more notches. It's going to be difficult, but I think there's a lot here. We know that additives are used to enhance products. So this right here is really rigid, but if I put something like a phthalate in it, it becomes really flexible. Again, it's those chemicals that have unintended consequences. We know that there's a lot of things used in food packaging and medical devices, which can leach out and go into our bodies. A lot of them have endocrine disrupting properties. We've known this for quite a long time. It's everywhere. And so really what we want to do is be able to measure these urinary samples using their mass spec and see, again, if we're making correlations between life history, concentrations of plastics in the body and concentrations of chemicals in the urine, which has never been done before. So this is a first. NYU is very bold and adventurous, as you could probably imagine. It's not NYU for no reason. They're also interesting at looking at these concepts of raising a child, right? 99% of the stuff that we're using in these situations are going to be plastic, whether it's a breast milk pump, a pacifier, a bottle. And we've seen some initial studies about something like a baby bottle can release up to 16 million plastic particles per liter of formula. And again, we don't know the consequences. We know that we've fully developed, most of us, as adults, but children, younger, we know from science, younger organisms just aren't able to handle this stuff as much. There's no large human study of microplastics and these endpoints that we've talked about here. NYU also collects data on fecal biometry, so looking at things like the head circumference, the femur length, they look at the birth weight placenta ratio, and they even look at the vasculature of the placenta itself. This is a whole nother angle that we haven't even started to go into, but we absolutely probably will. But that takes us back to my prior dilemma, and we need some money. The R21, it's a nice grant for where you're at if you're just beginning a project. It's for $250,000, which is a lot of money, especially in my world. And the R01 is specifically for the second step of this, which is, okay, you have data, but now you want to take it up to the next level, aim 2 and 3. This one is for $500,000. We actually submitted a proposal in February of this year for the R21, and we're doing February of next year for the R01. The success rate, they say it's 20%. It's not 20%. That's what they advertise, sure. It's probably more like 10%, maybe less. And I want to be fully transparent with all of you about what this process looks like. We submitted the R21 as a 74 reference document, 14 pages. And the first kind of step of this is to be assigned a percentile. Being assigned a percentile is kind of like when you say I was the top 5% of my class. You know, it's a good thing to be low. And this was kind of our first indication. You're waiting for a long time. You want to know, like, do I have a chance? Just tell me. If I don't, just tell me and I'll get kicked to the curb or whatever. So they assign you the percentile first. This is kind of what it looks like. So the percentiles are here. That's 0 to 15 or 20. The blue is funded. Green is unfunded. You can see that's a pretty broad range. I actually submitted an R21 with my old advisor, and we got 26, and that was a fast reject. Ignore the red. Actually, if you're colorblind, ignore it all. Sorry. This one is kind of a similar depiction. So this is the percent of these proposals that are funded. And again, the percentile ratings or ranges that you get. So 1 to 5, 6 to 10 look pretty good. It starts to go down 11, 16, 20. And again, I worked with a really well-known scientist. We got right here, big reject. In numbers, I know in the back, it's kind of hard to see this. If you get a percentile score between 1 and 10, you have a 99% chance of being funded. If you get 11 to 20, that goes down to 60%. And if you go 20 to 30, 9%. We got our number back. 9. Now it's not, I'm not gonna rest yet. You can see this little green guy here. That didn't get funded, and it was 5th. So you never know, but our chances of being funded on the first stage, 99.3 or something like that percent. So that's a big win for us. And the other thing that they don't really tell you is if you get the R21, you have a much better shot at getting the R01, which is nice. So hopefully, so we can say with certainty that over the next year, we're applying for, I think collectively, over $1 million of funding just for this. So this is a big deal. Very happy about that number so far, and we'll see what happens. I didn't advertise this, right? It's 2 full-time staff. Doing the placentas takes a really long time. I still got an email, and this group saying, hey, we heard that you're doing human tissue analysis and we're interested in kind of working with you. I'm a gastroenterologist. I'm really interested in seeing how microplastics might affect people with ulcerative colitis and Crohn's disease. Obviously, that's a big deal, and I'm automatically interested. And you might not have heard where this professor was from, Harvard, and I couldn't really turn down this opportunity. So I said, you know, write a proposal and let's see what we can get funded. And he did. He wrote a pilot study to investigate the role of microplastics in ulcerative colitis and Crohn's disease. Never really been done like this before. Now, what's interesting about this is we're not saying that microplastics are causing something, but it's an inflammatory response that you get from having something like plastic in your body. People that are battling ulcerative colitis and Crohn's disease can't really afford to have excess amounts of these inflammatory responses. So it may be that it's making it worse. He ended up also taking it a step further, and now we're not only going to get UC and CD samples, but folks with cancer too. All of this is incredibly important. It's never been studied in this capacity before, and we're doing it downstairs, which is really something else. So we're going to look at the associations between microplastics and those diseases, and we're going to see if inflammation is exacerbated in people that have microplastics in their system, and kind of use that to understand the pathophysiology of the disease as it relates to microplastics. Very excited about that. It's quite a bit of work, but it's really, again, the trailblazing aspect of it is pretty amazing for us. Now we talked about PFAS. PFAS chemicals, it's a lot of different chemicals. A lot of them have flame retardant properties. It's used in firefighting foam. Sometimes they use it to coat things. You've heard of the forever chemicals. Maine has a really bad situation right now with PFAS. A lot of the times it goes in the wastewater treatment plant into the sludge, and the sludge is applied on the land because it's really rich fertilizer, but that's where it has the opportunity to leach out. Became a really big deal here. And I kept seeing drinking water, well water, drinking water, but I kept thinking, where's it coming from? Where's the source? And so we ended up partnering with a really great company out of Illinois called Cyclopure. Cyclopure creates these tests that are very inexpensive that essentially give you the same data as something that's incredibly expensive, and it's really quick. So we did a little bit of a pilot study where we went around different areas of Maine, down east, and kind of tried to map out where we think it might be coming from. Is it the transfer station? Is it Penobscot? It was an interesting kind of first step for us, just exploratory. We didn't have a lot of plans for this. Then we started seeing headlines like this, really high PFAS levels in the Surrey Elementary School. PFAS chemicals have been shown to have bad health effects in people. So this is something that we're not theorizing anymore. We've demonstrated it. Michelle has a child at the Surrey Elementary School, and that started to hit very close to home. So we wanted to get involved however we could. So we ended up writing a very short proposal saying, we understand that you had high levels in your drinking water. We're happy to come in and do some testing. And then we can kind of generate a report, try to figure out next steps from here. It's been a wonderful partnership. They've been so eager to work. It's not always the case when you work with organizations where the more you find out, the worse it might be, but they've been so great to work with. We all got some nice press from it. Here's me in action. The really cool thing that Michelle is now working on is not only taking the data and populating a map with it, but you can actually highlight certain areas and it'll tell you the contamination or the concentration of these PFAS chemicals in certain locations. Really, really cool. If you get enough of these data points, you start to get a heat map of where it might be coming from. And we get a lot of kind of anecdotal suggestions about where it might come from. I'll get into that. We, again, got a nice little feature here. We love to see Shaw Institute's helping, yay. What we heard was that the soccer field was a dumping site at the Surrey School for sludge. So we wanted to kind of, again, help. We started partnering with a different company that analyzes soil samples. So Michelle and I went out there on a very frigid and again, sunny, that kind of rubs it in. Sunny and frigid day to collect some soil core samples that we could then ship off to this lab to look at their PFAS levels. And sure enough, we saw relatively high levels for us. Now, it's really difficult because we're at the kind of beginning stage of this being an issue. We don't really have a frame of reference for what's bad. And there's so many different classifications in drinking water, in soil, in a park. It's really difficult for us to say at this level, it's dangerous, but you just don't want to see any colors. The red and blue represent the soccer field at the Surrey School. So that anecdotal evidence might actually be pretty accurate. And when you combine all the PFAS chemicals, you get relatively high, again, in our experience, relatively high levels. So we started kind of doing this. It was a really great project because, I mean, this is like a case study, right? These are really technical terms that you're using with people that don't understand as much about science, but it's that opportunity to get together and be able to, us as scientists, work on our communication skills, and then for people to learn more about how we operate and how we can help. I don't know if you've heard, but in 2022, Governor Mills created this fund to support farmers who are really suffering as a result of this. A lot of sludge was deposited onto their land as fertilizer. It's contaminated with PFAS, so now they have crops with PFAS chemicals. They have the cow milk with PFAS chemicals. So the legislature appropriated $60 million to help with this. And it's called this PFAS fund, and it can be replenished at any time. And then they ended up hiring someone named Beth Valentine to be the director. She's a lawyer. They asked me to speak about the initial work that we had been doing in Surrey and around the area, the collaborations that we had. Now, this subcommittee that I talked to, it's just based on research. It's about making that $60 million accessible to not only farmers, but researchers. This group is not, it's very intimidating when I first started meeting with them. So you have members of the Senate, members of the House of Representatives, the dean of a UMaine department. And the coolest part to me is that they include a bunch of farmers, which is really great, because how can we be expected to make decisions for them? They need to have a place at the table. So surprise, surprise, a couple of weeks later, I get a letter inviting me to join the subcommittee by Beth Valentine and Governor Mills, which was awesome. And so since then, we've probably had 10 or 12 meetings trying to figure out how to make the $60 million accessible. How do you create a proposal program? How do you disseminate to the farmers? Can we buy a farm and use it to study the removal of PFAS, all those kinds of things. And I'm very, very lucky I've been able to work with them and kind of make progress. It was a lot of fun. I remember when I switched spots, like switched subjects, I have to drink. Something I'd started peak COVID, pandemic, whatever you want to call it, was looking into these things that I'm sure you've seen on social media, wherever else you see things. These laundry and dish detergent pods that dissolve, like the Tide Pods, or then now they have sheets. I love the packaging, right? The leaf, clean, it's very nice. 100% plastic free, eco-friendly, biodegradable, feel good story. I'll sleep well tonight. Ingredients meant to be small on purpose. Same thing with this one. I can't even read what this one says. USDA certified, safer choice, biodegradable. ingredients. The coating of both of these is called polyvinyl alcohol. It's plastic. I'm sorry. This is the part that sucks for me. Why is it okay for a company to put plastic in there and say plastic free, right? This says EPA Safer Choice Certified. So we started asking ourselves, well, what does that mean? Safer Choice Certified means that there's materials or there's chemicals that are given this association. If it's green, it's been verified to be of low concern based on experimental and model data. A half circle, kind of the same thing, but it needs more work. Then you get down to gray where it's not such a good thing. Polyvinyl alcohol is green. I'm not really sure why that is, but okay, back to our wastewater treatment plants and our poop that we love so much. What happens after you use a Tide Pod? Well, it goes in your laundry and then it goes to a wastewater treatment plant. Thankfully, there had been plenty of studies that looked at the behavior of this polymer in wastewater treatment plants. So what we could do, my co-author Varun and I, we could look at all of those studies, look at the degradation of different parts of the wastewater treatment process, look at how much of this material is used and project how much of it breaks down and how much of it doesn't. The claim of this industry is that it can be biodegraded in theory. So we do that. We set up an entire process in our paper where each part of the wastewater treatment process would break down PVA to a certain extent based on papers that have been published, and then stuff goes to ecosystems beyond. Well, we ended up looking at how much is generated in the US, how much goes to a wastewater treatment plant, might not. And then from there, there's a liquid that leaves and there's a solid. And the solid either goes to a landfill, it's incinerated, or it's land applied. Of the 100% of PVA that's used in the United States, 25% of it breaks down, 75% of it goes into the environment. Its behavior, we're not really sure. I actually scheduled a meeting with the EPA, that same group. I said, hey, that green dot says there's modeled and experimental data you have to say it's safe. Can we have it? No, it's protected. Can I do that? Can I be like, I proved this cool stuff in science, but you can't have it, it's protected. And I said, okay, so if you got data, then would that make you change what you're doing? And they said, no. And I said, you know that by you saying that it's that green dot, that people can lie and say it's biodegradable, eco-friendly, plastic-free, right? Well, we don't have any control over that. It's like, geez, the EPA. Thankfully, we got a little bit of media coverage from this, which was nice. Industry says they safely biodegrade. If you have a wastewater treatment plant where you train the microbes to specifically break down PVA, it can be done. The percent of wastewater treatment plants in the US that have that, 2%, 5%, nothing. And we don't know how it acts as a pollutant. I know it turns into a solution. I just don't know. In our paper, we never said it's a horrible material, it's super toxic. We just know it doesn't break down and it needs to be studied as a pollutant. Then the ACI got involved. The American Cleaning Institute is a multi-billion-dollar organization with 3,000 employees that got really pissed off about our study. And at the time, it was just 2 grad students. They end up writing this official letter to where we published our paper saying, oh, there's all these inaccuracies. They want us to withdraw the paper. And so the journal came back to us and said, will you address all of their concerns? Sure did. I love it. When you have 55 published papers on your side and they have garbage, it's really easy to go toe to toe with them. We responded and ended up being this official comment. And that was the end of it. So then they started putting out blogs. Attack on detergent packets ignores decades of evidence on film biodegradability. Sure. And another one, blog. I got a little bit annoyed about this, as you can imagine, because they're going after our characters a lot. And I went back to the journal and I said, whatever happened? You had them write a letter and then we wrote a response explaining to me what happened. We don't like to go into that stuff with the authors. Okay, can you explain the process? And they said, sure, we have an expert look at their response and your response and try to figure out who's right. Okay, what they say? We can't tell you. All right. Can you give me their critiques of our paper? And they said, sure, we can give you the critiques of the paper. They ended up going through that comment that I showed you, this one, and they said a lot of different things, but the best part was we would expect these authors to withdraw this comment because it had copyright infringement and plagiarism. Pretty funny, like you can't write this stuff any better. And so I said, can I publicize this and tell people this happened? No, we don't want you to publicize it. So I got really frustrated with that. And this is kind of where we're at now, but slowly we started to get a little bit more momentum. More companies kept kind of tying into this and saying, well, what's actually happening? They now make protein powder that you can take after you work out in the same pods. Now, it's technically not toxic. I think 98% of it you excrete and 2% stays in your body. I just don't want to be drinking plastic. It's not it for me. A petition was put forward then to say you need to change the safer choice list when it comes to this material type. The EPA shot it down again. But the best part of this is the more they got pissed off, the more they would kind of yell, and then we would get coverage in the Washington Post. So the Shaw Institute was in the Washington Post because this is still ongoing. So it's like, great, you just gave us $800,000 worth of marketing. Thank you very much. Keep poking. So this is still going on. We're actually in talks now with the University of Maine to do an actual research project and calculate how these particles affect certain organisms. And that'll be kind of revolutionary in this field, and it's going to create more pushback. But again, I like this coverage for the Shaw Institute, so keep going. So that's that. I know we're getting close to the end. So we've had a couple of recent publications this year. Michelle Berger, who was our associate scientist, published a paper that had been ongoing for many, many years, the gist of it showing how flame retardants are actually transferred from maternal milk to pups, and pups are usually not able to handle that type of stuff physiologically. Really important study. And then we zoomed out even farther and actually looked at kind of these same flame retardants in 3 different ecosystems, or 3 different parts of the world: Sweden, US, and the Arctic. We found these flame retardants in the Arctic, which you should not find anywhere near there. This is in review right now. Things are looking good. This one was published. We also published 2 more papers as co-authors. So there was a paper that we were on linking plastic pollution to climate change. A lot of scientific folks now are saying you shouldn't be studying plastic pollution. You should be studying climate change. It's worse. And we kind of say they're both related, very much so. At every stage of the plastic cycle, CO2 emissions are put out there. So they're very much related. And then there was another paper we just published on looking at using recycled plastic in construction. One of the other things that I kept, oh wait, this is the drink, Charlie, okay. I can't tell you how many times people would come in here and say, I've lived here for 20 years, I don't know what you guys do. Are you like a government secret lab or whatever, Area 51? And I kind of took that personally, because it's like, if we're doing all this cool stuff, I want people, bless you, I want people to know about it. I want people to see the lab. So we started small. We went to the farmer's market and put together kind of a makeshift touch tank with all of our information about the community sampling we're doing. And we talked to a lot of people there. That was a lot of fun. And then the Maine Maritime Festival, Steve's here somewhere. We're going to do this again this year. This is a rare appearance of Charlie the Shark. Now, Charlie the Shark likes to kind of show people that scientists can be fun and not so boring and, you know, uptight. He can talk to kids and make people feel better about science and that they can understand it really well. It ended up being a lot of fun. I'm super excited to, again, participate this year. Might do a dunk tank where Charlie the Shark can return back to the water, but we'll see how much pride I have. I don't have much anymore. We also ended up giving a couple really interesting and awesome experiences to the public. I was approached by the pastor of the Blue Hill Congregational Church, who was the mother of one of our former interns, about giving a talk there, and gave a talk. It was wonderful. We also participated in a beach cleanup with Plastic Oceans International, and that was just a huge hit. We set up at, thank you, Carter Nature Preserve, and had a booth, had a set. We did this in tandem with the Blue Hill Heritage Trust, which was a lot of fun. And we just went out and got a great group of people, kids, people of all ages, when, you know, incredible sunny, beautiful ecosystem. And I'm not bitter. Found all sorts of things that we shouldn't be finding there. But this is what makes it all worthwhile, right? Seeing this face. We ended up pulling out 40 pounds worth of plastic. And this becomes a really interesting step for us, right? Because now we have this new instrument downstairs. And this becomes a whole new experience, this whole new project to see what the material types are and maybe where it's coming from. Certain things, polypropylene is a lot worse than polyvinyl chloride. So it's important to identify what all these plastics are. And thankfully, we have this new instrument downstairs that can do that. Got contacted based on that new instrument by a group called Blue Ocean Society. Wonderful marine conservation group out of New Hampshire. And they said, we do microplastics cleanup. If we send you some microplastics, will you identify them? And they said, how much does it cost? It takes us about 45 seconds to get what the polymer type is when we're working on the instrument. So it's not super expensive. And it actually doesn't degrade the material, so it stays intact. The laser that it uses is actually less than this laser pointer in terms of the intensity. So sure, I did this report. We wrote it up for them. Nice kind of distribution of all the polymer types they're finding in New Hampshire. And then we hear about ladder. They had a 65-foot inflatable fin whale that you can walk through and see where the organs are and how big they are, which is just awesome. And it's based on a real fin whale who passed away. And they called him ladder because he was hit by a propeller and had a ladder shape on his back. So I kind of went, hey, World Oceans Day is coming up. Will you drive a ladder to Blue Hill and set him up? And I think maybe 2 weeks before the actual event, they finally said, we're so close, we're a volunteer short, and then we can make the drive all the way up there. The executive director of the entire organization ended up being that volunteer. And she drove up with one of their intern, or one of their, I think their marketing person, and they brought ladder. And this is ladder. The best part about it is that the Blue Hill YMCA ended up building this beautiful new facility. And we didn't want ladder to return back to the water if we had him outside. So we brought him inside. They gave us a wonderful rate to use this facility, and he got set up, and it was just the best. I never know how many people are going to go to things, right? You just hope. I thought I was going to get maybe 30 or 40, considering they agreed 2 weeks before the actual event. I check on ladder, ladder's all inflated, and then I go in the hallway, tons of people, tons and tons of people. They heard about it on the radio, they heard about it in the newspaper. I thought it was going to be like, we'll talk to one group and then they'll go inside and that'll be it. Nope. We ended up having to take them in sections because there were so many people. I expected to get 40 or 50, we got 300, which was really amazing. So we literally divided them up in between talking to the executive director of the entire organization, and then the group that got to go inside ladder. So we broke it up into pieces. And I'm sure you want to know what it looks like. I got a video here. There's ladder's backbone. This is inside the whale. I kind of fell there. And then heart, lung, blowhole, all inside. So this was the last group. It was a professor from Maine Maritime Academy, but it just ended up being the coolest experience. And it got, you put ads out for stuff in the paper. This actually got an independent story without us doing anything, which was really special. So we're going to keep doing events like this as much as we can for the community. Charlie the Shark doesn't discriminate of any age group. So we also like to go to Parker Ridge. I like to take whale bones that were given to us by Allied Whales so I can show the residents there the whales that I ate. So we've got humpback whale vertebrae, we've got skulls. I probably meet with Parker Ridge 3 or 4 times, every maybe 6 months, maybe a year, depending on how busy we are. Amazing group of people, super fun. And if this picture doesn't melt your heart, I don't know, they're holding hands watching Charlie the Shark Park. So that was the cutest. Charlie the human will also go over the summer with a kind of makeshift touch tank, and we'll bring crabs and fish for the residents to touch and hold, which is really fun. Even though we're close to the ocean, not everyone gets to make it down to the ocean. So this is a lot of fun for us. And they're always adamant that we come back and tell them whatever update we have. So Alma, who works there, she's on the call. We love you, Alma. We've been doing a lot of work recently with schools. So we had Brooksville Elementary, their 6th grade come. I gave a talk. This warms me up too, right? This is the hardcore engineering instrument that the 6th grader is using to understand plastic. Very, very cool stuff. We also work with this organization called iFuse, where if you do enough public outreach, they donate things to you. So they said, do you want a microscope? And I said, yeah. And they said, what are you using it for? I told them. They gave us 3. And each one of these microscopes is $300, 300 or 3,000. One of the two. So we now have all this donated equipment that we use specifically for outreach, which has been a blast. We also work with George Stevens Academy. So again, kind of that same thing where I'll put together a presentation, and then we'll take some folks down to the lab to do some lab work. And then we'll have, for example, our marine mammal bones to talk about marine mammals. To me, peak, like you made it with outreach, is if you can get high schoolers interested in something. And if you see this kid right here, I mean, that is a good face to have at a talk. And then we also work with Brookfield Elementary's 2nd grade. Charlie the Shark went and read books to kids about the ocean, which was a lot of fun. And then we also brought a lot of whale bones and whale skulls, and they were all over that. It was amazing. And last year was kind of us trying to figure out what works and what doesn't work and how can we form this. And so it's actually coming together pretty well, but definitely upping our community work game. We had Halloween at the Institute last year, and we must have worked like 60 hours that week, but we converted the Institute to being an underwater graveyard. So replaced all the bulbs with blue bulbs. And Charlie, the very passive aggressive shark here, would kind of judge people for coming inside his house. But we had a minke whale skull. We had a common dolphin skull. We all carved pumpkins. with marine species. Our gray seal was decorated very well. It was a blast. We really had a lot of people come in and interact with us. And again, kind of that same thing. What does the Institute do? And it was a great opportunity for us to share with them what we do. A lot of people came in to the course tonight and met a baby shark. It was very special. That was a lot of fun. And then met a dog shark as well, which was really cool. The other cool thing about our impact is we've gotten local politicians have been really involved. Obviously, when you're working on things like plastic pollution and PFAS contamination, the politicians want to get involved. So Senator Grohowski and Representative Milliken came by for a talk and a meeting. We've also had one of Jared Golden's representatives come to Blue Hill for a meeting, and it's a blast. Oh, you like that sweater? Pretty good, right? It was Christmas time, so it fit the vibe. And then here's the sun air holding a humpback whale vertebrae, which was very special. So I'm sorry I went a little long, but I wanted to thank you all for attending. First and foremost, Dr. Shaw and Cynthia for their unwavering support. All of our staff, this is it. This is us. This is our staff and then our interns, and that's it. We're getting all this done with just those people. A lot of amazing collaborations. Big shout out to the Blue Hill Wine Shop for helping to support this, as well as Momo's Cheesecakes, who gave us 2 wonderful cheesecakes that I hope that you have some of. And the Brick Block Jill was here serving as our bartender, which was incredible for us to have. And with that, I'll take a breath and I will answer any questions you all might have. Oh, yeah, Frank.
Years ago, I was involved in an educational program. The whole focus was really intervention and identification of young people with high-risk learning challenges. And we were in a cohort group of pediatric neurologists, a group from Harvard University, Tufts, Elliott Pearson School. And one of the things that came out very clear is a larger number of young people being identified with neurological issues, ADD, ADHD, or young people having the label Asperger's,
Absolutely.
autistic spectrum on a PDD scale. And one of the things that clearly came out is that much of the blame was being pointed towards some of the environmental factors, and they were using examples such as the fabric softeners you put into your dryer, and if they're not vented correctly, kids breathe in this stuff. And any thought about how much of this is impacting young people's cognitive processing, learning, more being identified with these handicapping conditions?
It's a really good question. Yeah. So the best that we can kind of do is to start by looking at how it behaves maybe in the environment with certain organisms like seals. And that research is hard by itself, right? Let's say that we find a certain chemical and a certain seal has a neurological problem. How do we tell that the neurological problem came as a result of the exposure to the contaminant? So there's a lot of challenges in the space, but to answer your question, we have demonstrated in the environment with certain organisms. It's very difficult. I'm only now getting into the human health space, so I was always doing other stuff. It's really difficult to do this research, but it's projects like these that can kind of give us that inclination as to whether or not, or how, at this point, it's how severe of an impact is it having on the physiology of humans. But I absolutely think that they are linked. And you can see certain conditions getting worse as we have higher amounts of these contaminants in the environment and higher amounts of exposure. Getting to there and saying, yeah, well, you might be able to say, you know, we have microplastics in this person has XY disease, but how do we prove that they're linked? You know, there's a lot of exposure all around us. The food we eat, the air we breathe, it's difficult to pinpoint it, which is why we haven't advanced it farther. But I absolutely think there's a relationship, and it's only a matter of time before we figure it out. And by then, you know, I hope it's not too late. It's a really good question that I wish I could answer better. Any other questions? I know I kept you all too long. Yeah.
Do you feel that you have a lot of public goodwill behind your standpoint on the issues, or is the corporate sort of plastic industry just as form of?
That's a really good point and a really good question. I don't vilify anyone. If the plastic industry wants to work with me on something, I will, because they have deep pockets and because they've a vested interest in this. I always try to avoid the language where you're saying this is bad, it's going to kill you, it's the worst. I sort of acknowledge that we need plastic at this point, but we don't need waste and we don't need environmental contamination. So it's kind of about meeting halfway and creating an environment where we can all sit down and share our collective issues, concerns, and figure out a path moving forward. So yes, there is support. There is a lot of pushback from both sides because I'm not going to vilify it and I'm not going to say it's perfect. I'm right in the middle. But I try to get people to understand that it's a really complicated problem. So it's never going to have an easy solution. It's always going to have a complex solution. But yeah, a decent amount of support. But as you can see from the other people, a lot of pushback in a good way. I think we were up against a pretty big obstacle in terms of taking what we're doing here and really being on that stage that where that potential was there. And it was tough, right? Convincing NYU, and I actually don't know how NYU and Harvard sounded or wanted to send us stuff, but being able to meet that, it's one thing for them to send samples. It's another for us to have the lab to be able to do it. So how do we in a grassroots way take the Shaw Institute in its current state and then be able to take human placenta samples? What do we have to do? So it took a really dynamic approach. And thankfully, I work with some incredible people that are willing to just wear 20,000 hats at once, which is what they do. And we're able to meet that. And I think we knew what we wanted to be all along. And it was just making it happen. That was the biggest challenge. But, you know, we're there, which is great. Any other questions before I pass out? But I encourage you to keep following us. As I said, we're making a new website. I'd love to show people the lab at some point. I come from a very instrument-based place, so we like to have a lot of instrumentation that enhances collaboration. I'm happy to give lab tours if you want to reach out to me or any of us at any point. We can show you around. Yeah. We have a question online. Okay, hopefully it's not my dad. Yeah.
Okay. To the best of your knowledge, what are some leading studies that solidify bioaccumulation of plastics in humans? I've run into people from the plastics industry that push the notion that there is no proof of negative effects of plastic bioaccumulation.
So we're more concerned about the contaminants, I would say, that are absorbed to the surface of it. But there are, I mean, I'd have to go into my computer and send the links. I don't know the exact studies. I've certainly seen studies where chemicals associated with plastics can get into any type of ecosystem at a lower level and work its way up. I'm happy to share that. If whoever asked that great question wants to send me an email, I'd be happy to send those papers. It's a great point that the industry likes to take, but we have seen ample evidence of the chemicals that are associated with plastics work their way up the food chain and concentrate as they do so. But that's a really good question. The first group that I showed you the poster in Ireland, right? The first group that came out to me when I was presenting plastics industry. And the lab that I used to work in was very plastic friendly. They like it, they just want it to be sustainable and green. So I was ready for it. It's like, all right, well, let's go, let's do this. And they like to have their little, you know, they like to have their standpoint, which is great. But we have plenty of evidence to support that. That's nothing new for us. Good question, though.
It's all about the sum of everybody.
It is. Yeah, we all have a part to play for sure. Any other questions? Yeah.
So you said that the state has allocated $60 million to help start with the remediation of PFAS and farmlands. In your perspective, is that a solid starting point in order to begin to address the problem?
I think the best thing about that plan is it's really diverse with what they're doing. It's not just remediation, it's exposure, it's source, it's replacement, it's conclusive, right? You can't, it serves a purpose. It's a flame retardant. We want to be able to give firefighters gear that's not going to catch on fire. So in its nature, it's a good thing, but it needs to be replaced, and it needs to be replaced with something that's not going to have these long lasting effects. It's kind of like plastic, right? Plastic's meant to last a long time, or this part, it's too long. It's supposed to be flexible, but then you have phthalates. So it takes understanding the source, working with wastewater treatment plants to see if we can remediate it there, working on cleaning up wells, having a replacement, looking at one farm that's clean from PFAS, looking at one farm that's contaminated from PFAS, and be able to figure out if we can use those as kind of experimental setups to see how we can improve our strategies for remediation. But it's a very dynamic approach. We can't just take it out. There needs to be something in there that has flame retardant qualities. But what? So it's very dynamic in that approach, and they're working on all of these different kind of parts of it. Remediation is definitely just one part.
It's like being a detective, you know, where you've got to sort of look at what the crime scene is, quote unquote. And sort of figure out, well, step by step.
Yeah, it's a slow process, but the fact that Maine is really leading the charge when it comes to PFAS contamination, it's been very impressive. I'm not surprised because our numbers are also not great. MOFGA is working on that as well. There's a lot of really great groups working on that. Awesome. Well, I think that's it. I really appreciate you coming. Again, remember, we have James Brown coming to talk on July 12th. That'll be a great one. All sorts of PFAS and plastic stuff going on there. He's much smarter than I am, so it'll be really fun. Otherwise, please tell us yourself if you like to take some cheese as you leave, or some cheesecake or chat, whatever you're feeling. Again, thank you for coming out. This was really great for us, and we appreciate your time.